Harpreet, Casey, and I are here to talk about meiosis. There are two full cycles in meiosis, but in meiosis 1, the first stage is prophase 1. In the beginning of prophase 1, DNA is very long, and it needs to be condensed into small chromosomes that are wrapped around histones. These come together and form bivalent structures, and once these structures are formed, they undergo a crossing over and a synapsis, which helps lead to genetic diversity and genetic uniqueness in their offspring. And you can also see how the centrioles and spindle fibers start to form, the spindle apparatus, and the nuclear envelope breaks down. Leading into prometaphase, the spindle fibers connect to the chromosomes, leading to metaphase. In metaphase, this is important because the spindle fibers align the chromosomes along the equatorial plane, as you can see here. This helps lead into anaphase. Anaphase is very important because, like prophase 1, this also leads to more genetic diversity because the recombinant DNA is now being separated and they are pulled to opposite sides of the cell.
This is followed by telophase and cytokinesis, which is the point at which the cells are actually separated into two daughter cells. And this occurs because actin filaments called the contral ring are formed against the inner membrane, and the ring actually contracts, which separates the cytoplasm of the two cells and forms into two. And here you can see there's two haploid cells that are created as a result that both contain unique genetic information, and this whole process helps to prepare the cells for the second part of meiosis. Oh, and the nuclear envelope is regained during that phase. And then in prophase 2, which is the first part of meiosis 2, the nuclear envelope once again is broken down in order for the spindles to have access to genetic information. And that's achieved in prometaphase 2 because the kinetochores, which are these protein masses, they help to connect the spindles from the centromeres to the chromosomes, and that is important because that allows the cell to manipulate it for the rest of the phases.
So after that, we have metaphase, in which the chromosomes align vertically along the equatorial plane, which leads us into anaphase, in which the spindle fibers have been shortened and the sister chromatids have been pulled apart to the sides of the opposite poles, and the centromere has been split, which leads us directly into telophase 2, in which the nuclear envelope reforms, and it goes into cytokinesis, which results in 4 haploid cells. And this is genetic diversity because you're going from the 2 haploid cells into the 4 haploid cells.